Hey Carp Geeks, so well, it's late November and in the US it's Thanksgiving next week, and for my sins I'm actually off to England to present at the Winter Carp Show. And what I thought I'd do is give you a preview of what I'm doing with my presentation, which is called Cracking the Amino Acid Code. So it's kind of an angler friendly version of the amino acid theory and how that's applied to baits. So as you can see, there I am on the title there. And so without further ado, let's talk through the slides. All right, so I'm going to kind of have my screen over here, so I'll kind of read through as we go. Okay, so obviously there's the website and the email if you want to get in touch. We pride ourselves on good customer service, and we love honestly talking to anglers. So if you got a question, feel free to drop us a line. Okay, let's take a peek. So, what are we going to talk about? Good question, right? What are we going to talk about? So, obviously, some very fundamental questions first. What's an amino acid, right? Because we use amino acids to attract and stimulate fish to feed. What are they, right? Okay, and then, a very important thing, a little bit sciencey, a little bit physiology based, right, is exactly how do fish detect amino acids, because if you can understand and crack that, you really understand how to apply amino acids. So we'll talk about how fish actually physically with their olfactory organs detect amino acids, and then once we've figured that one out, how can we then kind of use the theory to add amino acids to our baits and actually catch a bunch of fish. Right, so that's the trick. And then at the end, questions and answers. Now that's what I do in the presentation, and if you happen to be at the Winter Carp Show, feel free to ask questions at the end. And we'll also, I'll do an updated version of this, hopefully at the big one in 2025. Anyway, plenty of time for questions and answers. I do have a few standard ones people ask, and I'll provide answers for those. And as a kind of a bonus feature just for the video here, I got a few kind of comments on hydros, right? So people are always thinking about hydros and natural amino blends like you might get from chopped worm, for example. Talk a little bit about that. Okay, so let's have a quick peek. So what's an amino acid? It's a great question. So fundamentally, amino acids are the building blocks of proteins. There are 20 or so natural amino acids. You've heard of lysine and valine and things like that. So there are 20 amino acids. Okay, those form chains. So you get these long chains of amino acids, which then fold back on themselves sometimes to make uniquely structured proteins. Okay, so fundamentally, think of amino acids as Legos and the structures they make as proteins. Okay, now a little bit of a busy slide there, but what I want you to take away from this is that there are 20 natural amino acids, so 20 building blocks for proteins, and they come in 4 varieties. Okay, and that's key for later because there are actually 4 receptor sites which correspond to the 4 amino acid families. Okay, all right, so let's take a look at the next page. Oh, there it is. So my hero shocker, is Kevin Maddox, right? And as a teenager, I read Kevin Maddox Carp Fever in 1984 and was actually blown away by the amino acid study he did. And that's actually a direct copy from his book. Okay, so he saw excellent reaction, lysine, valine, okay? And then he literally ranked everything else, all the amino acids in order, and that order of reactivity, that order of potency, has held up through numerous since academic studies. So true scientific research has actually confirmed Kevin's findings from the 80s. So that was a fantastic piece of work and awesome, right? And what we were able to do is that literally build on Kevin's work and expand it. Okay, so let's take a quick peek, right? So there, remember I mentioned the 4 families of amino acids? Turns out that amino acids in the basic, which example lysine, and in the neutral, example valine, those are the 2 main families that cause the most stimulation in carp. Okay, so important details. Basic and neutral amino acids are the most potent, so they're great choices for fish feeding stimulants. I'm not going to get into the details, but if you read Carp Fever, he goes into a little bit more detail. The relative potencies of those actually cancel when they're mixed together, and that's the kind of fundamental kind of underpinning of what we call the BVRM, the bivalent receptor model, right? If you want to learn more about that, I'll flash up the webpage, aminotech.com. I've got lots of kind of sciencey type papers up there if you're really interested in the details. But take home message, basic and neutral amino acids, there they are, color coded in the table. Valine and lysine are the best, but honestly you can pick any of the purple and blue ones to make a carp attractant. Okay, moving on. So a bit more detail. Weird picture, right? So all living organisms, and these particular living organisms are really good at this. Okay, so we eat protein, and if we work out, we break down our proteins, our muscles, and then we rebuild them. So those building blocks to rebuild larger muscles are actually amino acids. This is why bodybuilders, and honestly, I get all my amino acids for my tests from health food shops where these guys get their, their milkshakes, right? So all living organisms are constantly breaking down their proteins into the amino acids and then replacing them. So there's a big turnover, okay? And if you do this kind of carefully, you can actually build muscle, right? Okay, but you don't have to build muscle to exchange your amino acids. It's happening all the time. Okay, now here's, for those of my age, right, if you grew up in the 80s, this makes sense, right? So there's a classic picture from Only Fools and Horses, and Trigger says, he works for the council sweeping the roads, and he says, I've had this, let's look at it. So I've maintained this broom for 20 years. It's had the same broom for 20 years. This whole broom's had 17 heads and 14 handles. So it's not really the same broom, right? And that's exactly how the human body is, right? It turns out that as shown by Trig there, right? Broom heads regenerate every 0.7 years and handles every 0.8 when you do the math, right? The human body actually turns over in terms of its cells every 7 years, right? So the person you were 7 years ago is not the person you are today. You're a completely new person, right? So all those proteins have been broken down and excreted and replaced, right? And here's the key thing. Every organism is doing this, right? And if we look at this picture here, it's kind of a weird one, right? So we, as humans, obviously are breaking down our proteins to make aminos, and we leave them everywhere we go, in our fingerprints, in our sweat, essentially, right? So this picture is an anhydrin, that's the chemical they use. It's an anhydrin fingerprint, and anhydrin stains amino acids, right? So everywhere you go, everything you touch, you leave amino acids on it, right? Similar with fish, they excrete amino acids all the time, which is almost like it's a tell, right? So it's like fish can detect amino acids. They can literally tell where fish are from their excretions, and they're doing it all the time involuntarily. You can't hide, right? You're always giving away amino acids, all right? So this is why we call it the primeval switch. This is why fish and other organisms have a very simple yes, no, food here signal based on amino acids, right? Which is an ancient kind of this is food receptor. Okay, so now this will get a bit gory in parts, right? So this is just pictures off the internet, not me doing things to fish, right? So obviously amino acids are detected through the fish's nose or nares into its olfactory system, right? So it's kind of interesting if you look at a fish's kind of nares there, they kind of have obviously an in hole and an out hole, and the out hole's kind of shielded, so it kind of deflects the water into the nares, which is kind of neat. Okay. Now that's the gory picture. So if you're able to dissect the fish and kind of peel back the skin there, right, you would see kind of tubes, and I'll kind of point them out with the mouse here. So these tubes are called olfactory tracts, right? They're basically tubes that then connect to these kind of bulbs here, and these bulbs are called the olfactory bulbs. Right? It's kind of a descriptive name. Okay, it's interesting, behind the olfactory bulbs is the fish's brain, which is only slightly larger, so the olfactory apparatus of a fish is almost as big as its entire brain. That's important, right? And then if we kind of like went inside an olfactory bulb, we'd see it would be a bit like a pomegranate. There'd be like a larger kind of volume, which you can see in the second picture, and then inside that larger volume, that pomegranate, are the seeds, right? And those seeds are called olfactory rosettes, and here's one in the third picture. Now, the olfactory rosette is kind of black and white there because that's a picture taken with an electron microscope. So we're getting super small at this point, okay? And if you think about stomach tissue, for example, the walls are kind of wavy, aren't they? Like the tines of a comb to increase surface area. That's exactly what's happening. Oh, pardon me, exactly what's happening down here. You can see the kind of the folds of skin in the rosette. Now those folds of skin are called the olfactory epithelium, which is kind of like nose skin essentially, right? And it's literally on that olfactory epithelium where we find the amino acid receptors. Okay, and it's kind of interesting. So we've zoomed in and imagine us to be sitting on the surface of one of these kind of folds of skin. On the surface of the folds of skin, it's almost like a rock down at the beach, right? We kind of see these anemones like in a rock pool, right? So there are these anemone type structures. You can see them there in the fourth picture. And they are actually nerve cells, all right? So they're kind of the stalks, if you like, of a nerve cell. And then emerging from that center hub is some fingers, right? Look like fingers of an anemone, and they're actually called cilia. And it's on those tiny little fingers, and you look at the scale there, 1 micrometer, right? So 1/1,000,000th of a meter is the size scale there. So on those tiny little fingers, that's where we find the amino acid receptors. Okay, so it's super small scale, and that's about as far as you can go with a microscope. Anything smaller is just a guess, to be honest. Okay, so next slide. Now there they are. So there's a zoom in on the kind of the cilia at the nerve cell. I've got a little bit of an animation to show you in a little bit, but let's go and take a kind of a quick look. So we're kind of getting into a stylized version. So there's the kind of the trunk of the cilia, and it turns out that the amino acid receptors, which are the black hexagons in this picture, are in clusters. Okay, and so clustered on these cilia fingers are the amino acid receptors. When amino acids bind to these receptors, it essentially creates a food here signal. So it's all about binding amino acids from the pond water to these cilia's receptors. Okay, now let's take a look at that cool little animation. So whether or not you get optimal stimulation depends entirely on the number of receptors, or I should say more accurately, the fraction of receptors occupied, right? And it's a bit like Goldilocks and the Three Bears. The porridge is either too cold, too hot, just right. And we've actually nailed down those regions pretty well. So let's take a peek, right? So we'll see a little animation here. Zooming in, there's the cilia, there's the receptor set. That's a low concentration of amino acids, and we get what we call a too cold signal, right? So it's less than 50% of the receptors are occupied. We don't get a good response. There is mild stimulation at best. It's not what you want. Okay, if I increase it just a little bit, we get just right coverage. Now it's shown in medical literature that drugs particularly work in what's called the therapeutic range, and we think this holds here too. And that therapeutic range of optimal kind of effect is between 50 and 85% occupancy. And that's exactly what we got here. We believe firmly, and we kind of have shown this by experiment, that if you're between 50 and 85% coverage, you get a just right optimal stimulation. So the fish are actively searching for food, not going crazy. Okay. Too hot. Believe it or not, you can overstimulate fish. Okay, and if that happens, the bites become very quick and you can't hit them. It's got the classic jacuzzi situation, right? So what you don't want is well, it's a bad word, but the technical term is overdose, right? So an overdose of coffee won't kill you, it just means you kind of like tweaked, right? So it's similar situation with amino acids. If fish overstimulate on amino acids, they simply just drift off. They become uninterested in feeding. So you want to avoid that, even though it's safe. It's not optimal for getting a bite. You want to stay in the Goldilocks zone, we call it, in the just right kind of coverage. Okay. Now, obviously, if you have a source of amino acids, like you sprinkle your bait with something, right? So powdered amino acid of some sort. As you throw that bait into the water, you start at 0. And then the amino acid concentration or amount of amino acids in the water grows, right? And so clearly that as that concentration grows, so does the coverage of the sites. And unfortunately, you very quickly go through that range of too cold, just right, to too hot, okay? Which has been, in my opinion, kind of the Achilles heel of amino acids for like decades, essentially, right? So there's our too cold, just to remind you, right? That's not going to give you enough stimulation. You increase the amino acid just a little bit, you get just right. Okay, and then it's very, very easy to go then with a little bit too much to the overdose condition, and that's what usually happens. It's usually under or over because it's very hard to maintain that just right coverage, and there it is. That's the major problem for amino acids. Okay, so I'll just read it, right? So you want that between 50 and 85% coverage. But this, in real terms, in terms of how much stuff you put in the water, is an extremely narrow range. So you need a very tiny amount, so it's all the way down there, right? And then that narrow kind of range within that tiny concentration is very narrow, right? So the actual numbers are right there. So I've used milligrams and a more convenient unit, grains, right? So if you think of a grain of salt, right? So you're looking at like 0.077 grains of amino acid per minute flux, so you got like literally 1/10 of a grain of whichever amino acid you like coming out of your bait to get the just right coverage. A little bit too much, you're too hot, doesn't work. A little bit under, too cold, doesn't work. So essentially this is almost impossible to do with pure amino acids or single amino acids, right? And that, as I mentioned, has been the problem with amino acids all through the years. I mean, we see they work in the tank, and then we go out and try and use them, and sometimes we catch, sometimes we don't. It's very inconsistent. Really what's happening is you're not getting it just right in terms of coverage. So that really is the take-home message. How on earth do you maintain the just right coverage? And spoiler, that's literally been 20 years of research, right? So I've been studying this problem for 20 years, and I'm happy to report that we figured it out eventually, right? So let's talk about how we figured it out. So how can we get that 50 to 85% coverage? Mission impossible. Well, not really. Let me show you how it works. Impulse, that's the product, locks in the perfect 50 to 85% by including a fixed fraction of blocking molecules, right? So what we do is, if we wanted a 50-50 coverage of blockers, which don't do anything, they just get in the way, and active amino acids, we'll just mix equal amounts and throw it in there. If you think about it, now it doesn't matter how much you throw in there, half the sites will be occupied with blocker, half the sites with amino acids, and you're going to be optimally stimulated all the time. And that's the genius of Impulse. It's now, as the key says, it's not about concentration anymore. You can use as much as you want past a certain minimum threshold. As long as you throw enough in there, you will get the right ratio, because it's a ratio, not a concentration. Hopefully that makes sense, right? That if you've kind of caught onto that kind of key concept now of ratio, not concentration, it's ratio dependent, not concentration dependent. And that really opens up the range of concentrations by about 3 orders of magnitude, right? So up to 1,000 times different in concentration from that single amino acid we saw earlier that's so hard to get just right. All right, so there we go. I told you my secrets, right? So have I got proof? Yes. Okay, so it's taken a while actually, but trying to get this to work in a tank is actually pretty hard because of the small volume. And as you can see from the slide there, the top right hand corner, it's kind of, I have 3 research tanks which I work with. I'm a chemistry professor, and my students and I have a research class where we study this stuff. And we have three tanks there, and they're set up in a particular way, which I'll kind of explain as we go through. But before we kind of start to talk about the details, I'll start the video going, right? So there are the fish, and that's optimal stimulation. So that's Impulse, just tested in the lab there. Okay, that's what you want. You don't want the fish going crazy. You want them actively searching. And if you notice, they're mouthing the gravel right around the doser there, which I'll explain in a moment. Okay, now this is not too different than Kevin Maddox's earliest work, right? So Kevin Maddox literally had the same setup, a little simpler. He just poured amino acid solutions into a tank and diffused them through the gravel. Now that was genius, because if you think about it, he's starting at 0, right? So even though he may have put too much amino acid in there, it took a long time to seep through the gravel. So he slowly ramped through too cold, just right, too hot. So he was able to see response from fish. And the quicker he saw the response, the more potent the amino acid. So very clever experiment, Kevin, there. Brilliant, right? So ours is just a kind of a slight upgrade on that. So we have a doser. So the doser is literally a kid's sippy cup with a hose going into the bottom of it with a, you know, an air stone from an aquarium to make it diffuse nicely, topped up with gravel, and we know the volume of that is 150 milliliters. Okay, so we then dribble in around 200 milliliters of amino acid solution through the tube, and we dye it sometimes just to see what it's doing, but after we've got the tank set up, there's no need to dye it. So that's kind of one where we did dye it, an early experiment, but it works just the same if it's dyed or not. So it's not about the dye. That's important, right? And as you can see, it's kind of acting a bit like a chimney, right? So we're getting a constant stream of amino acids into the doser, which then kind of bubble up and then tumble over the edge, right? And I've got it there, you Harry Potter fans, right? Godric's Hollow, right? So I've purposely put a little indentation in the gravel right next to the doser where the amino acids that fall out will collect like a puddle, and as you can see, the fish are hitting the amino acid kind of puddle there in Godric's Hollow. So that's kind of how you set up a tank, right? So very important. Tank setup is tricky because of the small volume, but it is doable, and these results are now consistent, right? So we get consistent results with this all the time, so we're able to do some pretty clever experiments. All right, so there's our lab proof, right? What about other proof? Well, back in the day, 2016, maybe earlier, I can't remember the exact year, but it was some time ago. Earlier kind of prototypes of Impulse we sent out to Sparsholt College for independent testing, and guess what? And you can look at the thesis at the Aminotech website. It came back that they were outfishing normal baits, otherwise identical normal baits, just one was fished with dosed with Impulse, one wasn't, by more than 2 to 1. So you can, you know, scientifically proven to better than double catch rates, and that's from Sparsholt. And there are actually 2 studies, one done by Tom and one done by Ollie. So guys, if you're listening, good job. Right. That was a while ago now. I know you both got careers and probably families by now, but great job, guys. Anyway, so that was Tom and Ollie back in the day. A bit more bang up to date, literally last year, as we really like nailed the formula, and it was super consistent every time. The results actually got better, and we sent it out for testing. So Luke and Ian from Carpology took it out and conclusively showed, and you can kind of search them on YouTube, just search the Carpology YouTube Impulse. They actually got a 13 to 1 catch rate ratio. It's kind of like insane. Luke was so tired at the end he could hardly talk, which I found the most amusing part of the whole video. But anyway, yeah. So Luke and Ian had a fantastic day, just bagging up on it, and so that was using obviously a big carp technique, 2 rods each, the usual thing. They actually fish with PVA bags, and then we sent it out to the awesome Mark Sawyer, great guy. If you ever talk to Mark, he's awesome, and sent it out for Mark for a match fishing style test. So he did a similar kind of thing, but with 2 kind of match method feeders, and he managed an 11 to 2 catch rate differential. So, you know, Sparsholt, Carpology, and Angling Times got some spectacular results, and they were filmed live, as Mark calls it, Jeopardy, right? So he just turns up and fishes, and people were coming up and watching him and stuff. There were no retakes. It was all as it happens, and those are the results. So pretty conclusive. Right? And so yeah, check them out. There's the Aminotech website if you want to take a look at it, and there's the Biosource Space website. Okay, now common questions. First one, it's more of a statement really, because it comes from questions. Impulse needs to get to the bottom, so you need to get to the bottom and create that Godric's Hollow type effect, right? So you need the amino acids to leak out the bait, saturate the bottom a little bit. Okay, so we use products that are either liquid or gel or paste. Those are our 3 types of product. The liquid is used with absorbent things like ground bait and pellets. The gel is used with hard things like boilies, for example. So it's, horses for courses. You don't want to dip the buoy in a liquid because it will just wash off. You want to kind of have a kind of sticky gel on it. Whereas a pellet where you can spray it and it soaks in a little bit, yeah, liquid's great, right? So horses for courses, obviously no spod mix, right? Unless you're putting in pre-dosed baits into a spod mix. All right. You got to get that minimum concentration. So if you remember the pictures, you need to get past the too cold, right? So as long as you put in enough, but don't overdo it, because there is a limit to this. Don't overdo it, but if you get to that minimum, you will start to get more bites. All right. Classic question, right? Now, I don't want to give my angling kind of colleagues, my business kind of colleagues, a hard time, but you see on certain videos, anglers dosing boilies for like months at a time and getting them to soak all the way in, and right, so yeah, and that's nice, right? And they cut it open and it's kind of soaked all the way through this kind of liquidy material, right? So the liquid soaks all the way into boilies, and you can soak them for as long as you want, and that's fantastic, right? But then that's a problem for us, right? So this doesn't work for impulse, and there's an analogy. So if you look at the picture there, that's a Norwegian fjord. And I've actually been to the fjords, right? And when you're on the boat, someone always says, how deep's the fjord? And it's hundreds, sometimes thousands of feet deep. And the person on the boat always gives you the same answer, and the answer is the fjord is about as deep as the mountains are tall. And that's a great picture there. So that kind of applies to my analogy of soaking boilies, because however long it takes to soak a boilie all the way through, that's how long it takes for it to leak out, which won't get you to threshold, which will not work for impulse. So with impulse, it needs to be a quick release, right? So it's absorbent baits with liquid, or hard baits with the gel, which kind of dissolves quickly off the surface once it's on the bottom. Okay, so there is a time component, right? There's a time component. Okay, last one. Amino acids are foods, right? Okay, so amino acids will be consumed by microorganisms. Okay, so if you're using impulse dose materials, use it as quick as you can. Okay, they're okay in the bowl because it's too concentrated to kind of have microbial action. It's a bit like honey in that respect. But once you kind of dilute it, yeah, the microbes can start to eat it. So try and use, if you do use impulse gel or paste, you know, try and use it as quick as you can after applying it. You know, it depends on the bait, but anywhere between 12 and 48 hours is kind of how much time you got after you've applied it to your bait. So it's a little bit of a different strategy. So you might want to kind of like put the gel on right before you cast in for a boilie, or if you're preparing a spod mix, you know, don't do it all at once, do it in parts, right? And so, you know, maybe make a couple of kilos spotted out, use that, and then make another couple of kilo fresh before you go out again. You don't want a lot of bait just sitting around, particularly in the summer. Okay. All right. Where can you learn more? Obviously, you know, there's the product range, and there's the QR code, and there's the website. We have a pretty decent website. You can learn all about the products and how to apply them there. Okay, if you want to send us an email, we've recently put together a really nice user guide, which we'll be sending out actually as hard copies with orders. And, you know, if you happen to be at the show, we'll be giving them out for free. Okay, so, you know, there's the liquid, the paste, and the gel. Again, horses for courses. Absorbent materials, ground bait pellets, the liquid. More harder materials are actually great for squirting into PVA bags is the gel. And then finally, you know, barbel anglers love it, right? The paste wrap is actually pretty good for wrapping baits, really good. Okay. All right. Last thing. Yeah, I'm a nerd. So long and thanks for all the fish, right? So for real. Obviously this is for the show, so if you happen to be at the show, yes, feel free to stop by the booth, pick up a book and have a chat, right? And obviously if you got any more questions, I'm happy to answer them at that time. Okay, now that's where if you're at the show we'd finish and we'd get into a question and answer. Obviously this is kind of a one-way deal. You guys can't really ask me questions, although if you want to put something in the comments, yes, I will get back to you, right? If you want to send me an email through the site info@biosourcebase.com, yes, I will get back to you. But I will address one thing which I mentioned at the start, and that is hydros. Okay, so people think hydros are the be-all and end-all of attraction, and honestly, they are not. Don't want to burst your bubble. Okay, they have limited stimulatory effect. Okay, what are hydros? Well, so you take a protein and you basically digest it, and that breaks it into all the individual amino acids. But the amino acids aren't there in equal amounts, so the profile, as they call it, gives you the kind of the percents of each amino acid individually, and they may go as high as like 7 or 8%, maybe as low as 0.5%. It depends on the food, right? So certain foods have more basic amino acids, for example, particularly meats, and vegetables tend to have more neutral amino acids, such as soy or something. Okay, and you know, here's A little comparison there. So, as we mentioned, each hydro has a particularly unique composition. So valine, as you can see, is really rich in tuna, lysine more so, and you just look at the numbers. Okay, there you go. All right, so each hydro is unique. Now here's the problem, and this is actually, you can take a trip back to 1984. Okay, this is why I was so blown away by carp fever. It wasn't just because of the results, but what Kevin actually said about combining amino acids, and there's his statement, right? So one conclusive finding was that those acids which were best on their own, guess what, lysine and valine, the two top tier aminos, proved to be ineffective when used in combination. That's fundamental. Okay, that's the foundation of the bivalent receptor model which we've developed. Bottom line is, you mix the two best ones together and you think, oh, that'd be awesome. They cancel, they don't work together. Kind of hard to get your head around that, isn't it, right? But that's how nature is. And what essentially any hydro will do, because the amino acids are in very kind of similar ratios, or similar amounts in the ratio, they'll cancel quite quickly. So you only ever get a very slight stimulation because of a slight difference in the amounts. So hydros aren't good because of the observation that Kevin saw. Now, my first project as a professor at the college when I started my research was to check that out, right? And here it is. That graph tells you everything you will need to know. So V is valine and K is lysine, and if you look right around here, a 2.25 to 1 valine lysine solution cancels exactly. Okay, so Kevin mentioned it, he saw it kind of with empirical observation, he was just observing fish behavior. We actually did an experiment. Yes, we were observing the fish, but we kind of recorded them and looked at them kind of after the fact, so we weren't biased in any way, and repeatable experiments. So you can see there, we call that the St. Olaf study, that yeah, you get a cancellation when you mix the two most potents together, as confirmed. Okay. All right, that's that. Hope you enjoyed it, and hope to see you at the show. Any questions, feel free to drop me a line in the comments or info@biosourcebase.com. See you next time.